said, I'm Cecilia Åsberg, and with me here as co-host is Tuija Thorvaldsson. So Tuija has the great, I think it's the great pleasure to introduce our speaker for today. Take it
Yeah,
away, Tuija.
it's my great pleasure to present Maya Hay, who is an expert on human microbe relations in food settings, holding degrees in dietetics, if I'm saying that right, food studies and communications. She's a postdoctoral researcher with the Center of Social Study of Microbes at the University of Helsinki, where she's in a little sister place that we admire very much from afar from the hub. And her research focuses on fermentation and the material practice of how we can come to know microbial life. In her current post, she examines the intersection of ferments and microbiome research, particularly as it reveals assumptions about what microbes are, how we can work with them, and how they get slotted into technosolutionist and healthist imaginaries. And we have also had the great pleasure of having Maya here with us on location in Linköping as a guest researcher for this term, which has been so lovely. And we are now very delighted to welcome her here today. So please, Maya, take the floor with your lovely presentation.
Thank you so much. What a great welcome. Ah, it's so lovely to be here, and I'm going to try and share all the things. Do you see what I see?
Yeah, I
Can I
think
get
we
a
kind of
verbal
see
yes?
what you see, but it's loading. Now we see what we should see. Yes,
Good.
perfect.
Okay,
Yes.
great. Thank you so much for having me. It's so lovely to see also some familiar faces and familiar names in the Zoom room. My first thanks goes out to Cece. Thank you so much for hosting me as a guest researcher at the Hub. It's lovely. I can't wait to see what we'll do together, all of us. Tuija, thank you for your support and guidance and for keeping me grounded. And also to Ombra, Jenny, and Laura and Carolina, who have also been such great peers and conversationalists during my time at Linköping. My title of both the talk today and for my book, my forthcoming monograph, is called Ongoing Song, Fermentation and Living with Microbes. And in both the talk and in the book, I try and take up what it means to live with microbes. Because we've already had, humans, mushrooms. Thank you, Anna. We've had human dogs. Thank you, both Donna and Deborah Bird Rose. But what of human microbes, especially microbes who make up and comprise and decompose our very being? And so I try to answer this question in my book, and I also try and theorize human microbe relations both now and ahead. And so I use fermentation as my method or my conduit for getting at and accessing microbes. And I'm interested in fermentation because it is a transformation both materially and discursively. So think of like grapes, right? The grapes turn into wine, and the wine gets symbolic meanings and values that do not associate with the grape. And that these processes of transformation also happen in one direction only. So you get grapes to wine, wine to vinegar, but you can never get back to grape. Or you can get from milk to cheese and cheese to blue cheese, but you can never get back to milk. Right? So it's always proceeding in one direction. And that relentlessness is, I think, a really productive and generative place to be, especially when trying to understand how to work with microbes, because these transformations are made possible by both the metabolic labor of microbial life, but also the reproductive labor of microbial life. And it so happens that out of all of the food transformations that can take place, fermentation has spanned all food cultures, and it's also a process that we've relied on for millennia. And also there's increasing research that shows that we're not the only species who ferment, which I think is quite fascinating and generative. And so far, most of the studies that involve fermentation have been focusing on the fermented food products. So what does the wine mean, and where does the wine come from with terroir and the IGT labeling, DOC, DOCG, DOP, AOC, various labeling, or it looks at the cheese product and the sort of regulations that it faces, right? And I'm interested more the process, and I'm interested in the process because it involves microbes through and through. And so the why microbes question is something that I get often. And I'm interested in microbes because, as Sander Katz, who is a fermentation revivalist, a self-taught queer fermentation revivalist down out in Tennessee, he says that microbes are everywhere always. And they also have been, right? Microbes have been everywhere always. It's just that our attention to them might be a bit more recent. And it's the term microbe is actually quite a lazy term, because unlike other forms of species that we sort of categorize, microbes, which involves all kinds of organisms, are only really defined by their size, that they're too small for us to see, and that this invisibility is actually a human limitation. It's not really a defining feature of them. And microbes span anywhere from bacteria, molds, yeasts, viruses, protists, archaea, amoeba, some algae, and then some. And because they have been invisible, quote unquote, for the most part, of humans writing and studying them, we've also been taking microbes for granted, so much so that we've really not theorized them, especially in terms of who and how they become part of us, or how we become part of their worlds as well. And the part that has been theorized is usually along binaries, and the binaries of good and bad, that sometimes they're good for us, sometimes they're bad for us, sometimes they help us, and sometimes they harm us. And this is where I really rely on the work of feminist theories and feminist philosophers who really pick apart binaries, because the very fact that they're a dualism, or the very fact that they're a binary, guarantees their ontological separation. And I'm interested in more where and how these sort of overlap and come together. And the last part is that at least until quite recently, how microbes have been studying has been primarily been confined to laboratory settings with capital S science. And part of what I'm trying to do, both at the center that I'm at and also in my research career, is to try and break those boundaries a bit and to try and develop other ways of studying microbes outside of laboratory contexts. And so combined, I'm interested in the question of, well, how do we live with microbes, and really sort of frame the question of living with versus living off of. And this is really consistent with the sort of Anthropocene, post-Anthropocenic question of like, what does it mean to live with these other beings on this burning, damaged planet? And fermentation, in particular, allows me to study what it means to be human on a highly, highly microbial planet. And this question of, well, what does it mean to be human, really aligns with the sort of post-humanities destabilizing of the human, the microbe, the environment. These are not stable entities and never have been. But the post-humanities toolkit really allows to kind of play with that destabilization and kind of see what comes of it, especially when these are constantly shifting and becoming. I'm also interested in the living gerund, the verbs that are in different tenses, because not only are we talking about the present living with, but also, and here I am inspired by, for instance, the work of Rosie Braidotti and others who really think about the future anterior. What will have needed to happen now in order to make such futures possible? And it's really the actions and the practices that are ongoing and happening now that really allow for that kind of future to come about. So I'm not really interested in fermentation as this like stable noun or this static object or a static phenomenon. I'm really interested in the dynamism that kind of keeps and feeds its change. And then this also has methodological implications of how do we study something that is constantly on the move. So I'm not interested in microbes as a snapshot of like, well, these microbes were once there, right? We can take that snapshot to mean this is the microscopic thing that I see in a microscope right now. We can also consider the snapshot as like what are now used, which is the metagenomics, which are like genetic traces of microbes that were once there. I'm not interested in the snapshot so much as I'm interested in the perpetual encountering of microbes. And methodologically, that means being immersed in microbes. It means also being influenced by various strands, such as new materialist thinking. And here, I was trained in North America, so I align more, not align, I reference more the sort of Diane Kuhl and Samantha Frost more than the Iris van der Tween. But I'm also interested in things like embodied knowledge and just different conceptions of the body, the unbounded body, the sense-making body, the sensing body. I'm also interested in the more than human and the multi-species entanglements. And speaking of entanglements, I'm more interested in sort of feminist science studies, feminist technoscience, agential realism, and how things even come to be known as such. And so then this leads me to the questions of, well, who can know how and to which effects? And so with this, I ask this question for the book. What are the hands-on material practices of fermentation, and how can these practices help us to understand how to live with microbes, but also how to continue to live with microbes? And it's this futurity and ongoingness in mind. And so practically, this means how do people work with microbes? Like what are the actual hands-on moment-to-moment decisions? But it also means the ontoepistemically, like the ontoepistemics of the question, how do people know what microbes are and how do they know what they know? And then also I have ethical questions of, well, what does it mean to continue to think and do life with microbes, and how do we do all of this in a highly microbial world? And folks who are familiar with Baradian agential realism will kind of notice that sort of tripartite, the three-part onto-ethical epistemologies that are always there. So this is this book is an ethnographic account at a Japanese sake brewery. And sake, for those who do not know, is an alcoholic beverage that's made entirely of rice. It has actually three main fermentation processes that then make up sake. So it's not just one fermentation process from one thing to another, but it's actually three steps that are woven into one. And I framed the brewery as a different sort of lab and sort of a different way to understand how knowledges get produced inside this brewery. And I collected data that spanned 2 brewing seasons, and a brewing season is a winter, usually from about October to about March or sometimes April, depending on the year. And during this time, I worked as an apprentice brewer. And this, by doing the work of brewing, I was able to both work alongside brewers, but also do the work firsthand. And this was very important for me to understand and stay committed to those hands-on material practices, those moment-to-moment decisions that happen over time. And it also allowed me to move with the research. And this movement becomes a very pivotal part, both in my title, but also in some of the conclusions that I draw. And I really wanted that immediacy of participation. And an example of that participation and sort of that movement, as I sort of alluded to before, is relevant, or it shows in my title. And this ongoing song is not just a metaphor. It is also referring to a song that the brewers use to keep time in one of the brewing steps. So this is just sort of one example. They have another song at another step, but I'm kind of using this one and showing it as a quadriptych at the moment. The steps themselves are just mashing back and forth. It looks a bit like rowing. But the song is used as an alternate form of timekeeping. It's an embodied form of timekeeping. So rather than have like a stopwatch, right, that says, okay, and now we do this motion for the next 12 minutes and 37 seconds, it is performed through the song and through a number of verses. The song itself has 15 verses, but it is not every day or every time that all 15 verses are performed. And this is where the head brewer's expertise comes in. And it is up to the head brewer to make the calculations based on the ambient temperature of the day, the state of the rice that got steamed, the temperature of the rice itself, because all of these and many more, because I'm not a head brewer, I don't know the full calculation, all of these different factors then affect how long this particular step happens, this mashing step, and performed through the song. So sometimes the song is performed up to 10 verses, sometimes 12, sometimes 15. But the important part is that because it's something that's embodied, you can feel when the song is too fast. You can feel when it's too slow. And so it is through this embodied notion of time and processing that then get repeated over time, that then they have a certain frequency, they have certain sequences, and it changes during the brewing season that I really started to pay attention to microbial temporalities. It's also a very performative song in the sense that it's an enunciation that materializes change. Right? You start with one thing and you end up with another. You start with rice and you end up with, in this instance, a starter culture. And so by looking at this microbial time, I came to the conclusions that fermentation doesn't begin and end when a human says so, which might seem really obvious, but I think we tend to clutch on to that human-centric and human exceptionalist understanding of, well, of course, things only happen when we preside over them. Not always the case. And that because microbes are ubiquitous, because they are everywhere, it means that the possibility of fermenting is also always there, and that the possibility is also quite latent. And so here I'm going to read a bit, an excerpt from my introduction, because I think it captures some of this contribution in a neat package. So I frame fermentation as an ongoing practice because doing so helps me think about themes like continuity, maintenance, and the specificity of care practices, including when and how to intervene. The shift from linear process to ongoing process relies on the fact that microbes are everywhere and latent, ready to act when preconditions are met. It's a subtle shift, but an important one, especially given the long history of obliterating microbes wholesale. Obliteration is the antithesis to ongoingness, and it is a myth. Having a so-called upper hand over microbes is ever only temporary and thus aspirational. And here, just look at AMR, just antimicrobial resistance or novel viruses to really see the myth of human control. And rather than pit our living at the expense of microbial dying, I am compelled by both scholars and practitioners who trouble this accounting of certain lives like an expense ledger. And so this idea of what we do and how we intervene is important, but I think the ethnographic data really brought me to when is just as important as what is being done. And this timeliness is really crucial. And this is, I think, how and why I've brought this movement and choreography and this language of dance really into the monograph. So here I talk about microbial choreographies and syncopations and rhythms and syncing up with and being in step with with microbes and their metabolisms. And again, I'll read an excerpt from my introduction. Our futures depend on how we learn to tune in to microbial temporalities. By focusing on the making of ferments, I show the different tempos, rhythms, sequences, frequencies, time signatures, repetitions, and cycles that texturize microbial time in fermentation. Relatedly, I analyze how brewers learn to keep in step with microbial time as they adjust their entire work schedules and task lists around microbial activity. And this entails practical questions of trying to assess what microbes are doing, as much as it raises epistemological questions of how brewers come to know microbes and their timings. And because the brewery handles a range of microbes, I account for the temporalities of different bacteria, molds, and yeasts, and show how they sometimes align with our time, most of the time not, in the longue durée of fermentation. And then I am curious also about the ethical implications, and this is what I hope to share with you towards the latter part of my talk, which is about moving from the hyphenated responsibility towards the hyphenated adaptability. And I'm proposing that fermentation shows us how to adapt to the unforeseen and the unknowns that are ahead. So that's a sort of snapshot of my forthcoming book. It's with Minnesota Press. I have 7 chapters, and I'll be sharing a part of my conclusion in the last chapter on improvisational ethics. And I'll briefly give a one-liner or sort of preview to each of the chapters, but first I'll give more details about my field site, which is a very particular brewery. So Terada Honke is one of 2 natural sake breweries in all of Japan, and at the moment there are a little over 1,000 breweries in Japan, and of the over 1,000, Terada Honke is one of 2 who brew naturally. And while I'll go into more detail of what that means, naturally brewing, in a moment, what's important for understanding this field site are a few different details. As I'd mentioned prior, brewing happens in the winter season, as you can see from the snow in the photograph. And what that means for this part of Japan, which is out on the Chiba Peninsula, it's about an hour and a half, maybe, east of Tokyo. It's right on the sort of coast of the Pacific. And in this part of Japan, in the wintertime, the temperatures are about -2 to about 12, +12. And that window happens to be quite good for brewing this particular style of sake. And Terada Honke has been around for about 350 years. And most of the brewing processes are done by hand. And so that requires a lot of coordination amongst the about 8 to 12 brewers who happen to be there for a brewing season. And when I say most things are done by hand, there are really only 3 machines. There's a crane that lifts a giant steaming basket. There is a forklift that lifts giant pallets of rice. And then there is a machine press that presses out the liquid that allows, that separates the liquid sake from the sake lees, which is the solids. Everything else is done by hand, meaning all of the schlepping of pallets and pallets of rice is done incrementally, usually in increments of about 10 to 30 kilograms at a time. And so you can imagine that there's all kinds of synchronizing and coordinations that have to happen amongst the different brewers. Uniquely, this brewery relies exclusively on ambient endogenous microbes. And this is, I think, the key aspect to the natural sake brewing. So rather than add a vial of bacteria or add a vial of yeast, what this brewery does is it relies on the microbes who are already in the brew house and the brewing environment. And so I'll show 2 more photos to kind of give you a sense of what that means. So the endogenous microbes live on the walls, the crevices, the tools. There's some in the water, there's some on the rice, And so the work of brewing not only becomes about coordinating between brewers and sort of processing incremental loads of rice, but it's also about coordinating with the microbes that they cannot necessarily see. And so the work of brewing becomes gathering very specific microbes in a certain order at a certain time in certain steps. And this is all, like, to me, was very fascinating to be able to figure out, how do brewers know what to do when? And so this is the key thing I want to point out in this photograph is the giant entryway there on the side of the photograph. That is the main entryway into the brew house. And the brew house, as you can see, does not really have a door. There's no rolling door that you can't see in the photo. Like, this is it. This is the gate. This is the opening. And so I show this because what is inside is outside and what is outside is inside. This idea of interiority and exteriority really fall apart in this particular context. And so this is now the same photo taken from the other side of the gate, of the entryway, right? And so this these processes that take place in the incremental steps of brewing are really relying on microbes who are everywhere all of the time, but really try and target very specific ones at certain times. And that is the work of brewing. So in chapter 2 is when I go through the different steps, and I call this the many steps of brewing and the microbial choreographies of the many steps of brewing. And I describe it as a choreography because there's all of these sort of synchronizing and coordinating that happens at different times. It all has to kind of come together during a brewing season in order for anything to happen. In chapter 3, which is called Degrees of Knowing, this is my epistemology chapter on how brewers come to know what microbes are doing. And here I propose, I look at how brewers measure different things over the course of the brewing season, and I focus specifically on how they measure temperature. And I come to the conclusion that they are okay with a certain threshold for knowing, and I call this knowing enough. That knowing enough is good for this particular brewing setting because it takes the time that it takes to measure something takes time away from doing the thing. So there's a threshold for them to, like, this is good enough, we'll move on, because moving on is more important than understanding perhaps the specificity or the precision to knowing. Chapter 4 is about embodied attunements. This is more about embodied senses, embodied knowledges, but it's also about the song and the sort of lyrical enunciations that go into the song and how they not only are performing, but are also performative in actualizing and materializing human microbe relations. And here I focus specifically on proprioception, and I try and propose this idea of microbial proprioception. And just as a fun juxtaposition, this photo in the very center, in the very, very back, is a very small shelf. It's called a kamidana. Kamidana is like an offering. It's a small shrine. And just to give you a sense, I'm about to change the slide, and it's the exact same angle from which I take this photograph. But in this one, you can see so much steam coming off of the rice, and the steam is so much so that you really can't see past. The bellows of steam are so much that it's difficult to see past even a few meters. And so what that means for the brewers is, again, they have to find ways to coordinate with each other through vocal call and response, because we can't necessarily see what's happening with other steam, or because it's, things are happening in another part of the brewery. So this, again, at the top, you see the kamidana offering, and chapter 5 is kind of focusing and takes inspiration from that offering shelf. It's a religious offering or a spiritual offering stemming from the sort of Shintoist ideology or cosmologies. And here, the chapter is called Naming the Unknowable, because I'm trying to understand how brewers name microbes and sort of dwell in the fact that they have difficulties naming and precisely naming what microbes are, and they sort of dwell in that indeterminacy. And I really call upon here, instead of ethnographic data, I look more at interview data and sort of look at, well, what do they call microbes? How do they characterize them? To what extent do they anthropomorphize them? To what extent is the language animated? To what extent are microbes thought to be animated? And sort of juxtapose all of that in the context of Buddhist and Shintoist understandings of the world, of the natural world especially. And then chapter 6 focuses also on a different data set. It's looking at a book that was written by the late president of this brewery. He wrote a book about the path of fermentation, what I learned from microbes, which to me was like too dead on to not take up as a data source. And even though the chapter focuses on this ethos and affect of playfulness, I sort of trace the playfulness back to this book and what that there's a reason why this late president put playfulness in place. There's a reason why he called upon playfulness to understand like how to work with microbes. And then I trace it back to the ethnographic present to then show instances of playfulness at the brewery while making sake, and sort of tie it to understanding and being okay with microbial uncertainty. And it's this uncertain element that then leads me to my final chapter of improvisational ethics and dealing with microbial uncertainty. And so, why improvisation? Improvisation is a way to not only create the unforeseen. This is, if we think of like improvisational jazz or improvisational dance, it's a way of composing something that hasn't been scripted before, but it's also a way of adapting to the unforeseen. And so we can think of like, yeah, I had to improvise because I ran out of something when I'm cooking, right? Improvising a recipe, or like improvising in a business meeting or in like a spoken word, public speaking engagement, right? So this idea of improvisation has really allowed me to pick apart how is it that we deal with the unforeseen, and that how is it that we can improvise certain futures with microbes. And I build on this idea of improvisational ethics. I build on these ideas using a political theorist's work by the name of Emily Beausoleil, who discusses dispositional ethics. And Emily Beausoleil's work, while wonderful, really focuses on human-human interactions. And sort of, Emily Beausoleil arrives at the conclusion that and therefore we must listen better. And then it got me which is true. Yes, we must, we should. But also, what does it mean to listen to microbes who we can't necessarily easily heed? And so that's really what brought me to how is it that we can respond without being able to hear? And so then that got me to my current understanding, or my current sort of conclusions of moving from responsibility towards adaptability. And I am hoping to read you in the time that I have left some of the work that maybe describes that adaptability more keenly. And I'll maybe start with here. All right. This is an excerpt from my conclusion. Multi-species and post-humanities discourses about control and care are ripe for discussions about power relations with microbes. For what are we to do when nested crises are imminent and underway? Key voices in this conversation include Vinciane Desprez, who calls for a self-reckoning with all too human assumptions about what kinds of questions we ask of other creatures. Desprez calls for staying open to surprise, of making the reflexive verb fall back in on itself in phrases like I interest myself, je m'intéresse, when encountering other beings. But these voices also include Haraway's formulation of a hyphenated responsibility, wherein cultivating the ability to respond and respond well is prioritized over being responsible, as if it's a box to tick, like been responsible, done that, or as if it's a status to unlock as a card-carrying member for life. I'm responsible. I am this way for life. And both Dupré and Haraway describe ways of learning how best to respond over time, and both work towards the renewal of becoming through deep familiarity with the animals under study. And this is important work, but I'm also interested in like beyond thinking about what to do and then asking, what do we do? Haraway prompts us with whether we, this is a quote from her, whether we asked for it or not, the cat's cradle pattern is in our hands, so think we must, end quote. And later she repeats, quote, we must think, end quote. And so this repetition of think we must think. And I tilt my head at thinking somewhat because I'm trying to point out every aspect of our entanglements with microbes who are everywhere and always. And I'm trying to account for every strand of every interconnected species, of locating new areas to study in anticipation of some deluge that interrogates, well, what about this species? Or what about Y species over there? And we're saturated with capacity now. And so I think that we are capable of response and we just choose not to or can't for some other reason. And perhaps that it's time to shift energies and not paradigms towards rendering oneself capable of deciding what to do. And so it's this bridging of theory and practice that leads me to fermentation because it's one way to improvise a future with the unknowns in mind. So if fermentation shows any way forward, regardless of the sort of intrigue of food or the intrigue of species, it's that fermentation tries anyways, even in the face of not knowing in full. And with incremental changes, fermentation serves as a way to practice adaptability in microbial worlds. And again, there's no guarantee, there's not knowing that this will work out, and we're not fermenting towards the utopic or improvising towards the just and restorative. And to improvise takes seriously the structure of provisos and the contingent relations of all the different human and more than human beings, and the potential to adapt in the face of the forever unseen. And this is what I mean by practicing adaptability, because past the call to consider responsible relations lies the work of practicing it and practicing it in an ongoing matter. And this is where I start to wonder, and how this is where I start to also try and expand away from food fermentation towards other microbial transformations. I wonder, how can we take the basics of improvisational ethics, which are inspired by fermentation, and use it to understand, for instance, choreographies of containment in the hospital or activated sludge at the wastewater treatment site? What are the time signatures worth noting in specific encounters with microbes? How can we make sense of them otherwise? We seem to be able to, for instance, hear the difference between the bumblebee, a fly, and a mosquito, but we can't seem to tell a difference between bacteria and yeast. And more importantly, how will we know if we're right or wrong? Because this uncertainty haunts us. We are terrible at holding on to uncertainty. We are even worse at keeping the world ambiguous. And we are the worst when it comes to both uncertainty and ambiguity in the context of microbes, because we're constantly under the siege of what ifs. We have what if questions festering in our minds while the mother lode question hovers over us like a cloud. But how can I see the proof? How do I know for sure? And of course, these are caricatures of questions, but my time at Terra de Honke taught me how to know microbes otherwise and bask in multiples. We also have a tendency to eschew binary constructs in favor of monorealities, but perhaps there's also a value in seeing ambiguity as an empirical process, to name it as such, and to try and articulate the various levers that pull microbes into view, and brought into view in a particular way at a particular time. And to overwrite this ambiguity and tidy up conclusions into a neat do this, not that, is to miss the glaring challenge ahead, that microbes are both everywhere and nowhere, living and dying because of what we do and don't do. And this inexactness is precisely our problem and our salvo. We are always and already in contingent relations with microbes. We cannot disengage from them any more than we disengage from the air. Plus, microbes have figured out how to persist in the face of obliteration. We have not yet, but our thriving does not ensure theirs. Remember, microbes can live just fine without us. So by asking what kinds of futures we want to improvise with microbes, I'm trying to highlight a middle road between hype and doom for what lies ahead. To think of fermentation in terms of an improvisation can make it easier to navigate options because, in its ongoingness, there are plenty of opportunities to iterate, fail, and try again. So, regardless of whether one makes ferments in the home, cracks open an egg, or feeds a pet from the side of the table, there will be microbes, and with it will come opportunities to decide how to engage with this invisible register. Thought of in this way, humans can sometimes act as a catalyst in microbial improvisation if and when they create the conditions that are conducive for microbes to do the work. The antithesis is stagnation, and how we engage or not has implications either way in the microbial world. And rather than be paralyzed by uncertainty, the trick is to stay adaptive and attuned to the rhythms in the moment. And that I think, be my cue to wrap up. I will maybe just say two quick last things. The rest of the chapter is really about how no one can claim neutrality, that they can't say, well, I'm not a fermenter, or I'm not a brewer, so this doesn't matter to me. I say that, well, we're all co-implicated. Microbes are everywhere, so what we do or don't do actually matters, whether we're in a brewery or not. And then the other thing is, thank you and stay tuned. This book will be coming out soon, question mark. Likely next year, if I'm lucky.
Wow. Thank you, thank you, thank you so much, Maya. This was amazing. You are such a both writer and speaker, and I'm just like, wow. This is fantastic. To give everybody the chance to really, collect the thoughts and think about what they want to ask you. We are now gonna stop the recording. Yes.